Hi friends, good morning to you all. So in this today's lecture, we're going to start second half of our syllabus, that is food microbiology. So in our previous lectures, we have already covered dairy microbiology. So in this today's lecture, we're going to see how microbes affect this area of food. So microbes affect the area of food as like milk or dairy industry. In case of both of these things, microbes affect positively as well as negatively. In case of a positive approach, we have seen in milk microbiology that there are so many products that are fermented by different types of microorganisms, and these milk derived product, these are derived from different types of microorganisms, just like cheese, dahi, yogurt, etc., all these things, and we consume this product on daily basis. If you consider negative approach, so there are some microbes, especially there are some microbes, they causes spoilage, as well as there are some microbes, they cause especially pathogens, they cause various milk-borne diseases. So in the similar way, food also we have to study microbiology with respect to food in both positive as well as negative way. So here in this today's lecture, we are going to see basic aspects of microbiology that is related to area of food. So food science of basically food science, it is a branch of agriculture science, and it is quite different from the field of nutrition. But as far as food microbiology, so the food microbiology is a very broader one, and it encompasses the study of microorganisms which have both beneficial and deleterious effects on quality, so the field of food microbiology is a very broader one, and it encompasses the study of microorganisms which have both beneficial and deleterious effect on quality and safety of raw as well as processed food. So microbiology affect the food, both raw as well as processed food. So it is important to understand a relationship among the various microorganisms making up the microflora of food. Okay. So we should know which type of microorganisms they are commonly present in food and their role in spoilage of food as well as other activities they carried out in food. So in fact, food microbiologists are concerned with practical implication of the microflora of food and food microorganisms that can causes spoilage of food and diseases in human. So the primary tool of microbiologists is ability to identify and quantitate foodborne microorganisms. However, the inherent inoccurrence in enumeration process and natural variation found in all bacterial population, it is complicated job for microbiologists to enumerate exact number and exact types of microorganisms. So moreover, they may be important from aesthetic point of view. Of course, some useful bacteria may be important because they change the functional properties of food stuff, resulting into new taste, new odors and new texture. So microorganisms which are present in food, it includes bacteria, molds, then yeast, then algae, then viruses, then parasitic worms and protozoa. So these kind of microorganisms, they are basically present in food. So they includes various types of bacteria, then different kinds of mold, then yeast, then algae, then viruses, then parasitic worms and protozoa. So these organisms differ in size as well as shape and in their biochemical as well as cultural characteristics. So what we have to do, we have to study which type of microorganisms are present in food and what are their role. Okay, so the microorganisms described below are among the most important genera and species normally found in food products. Okay, so first we'll see bacteria. So each microorganism has its own particular nutritional and environmental requirement. Okay, so in that we're going to see first bacteria. So First is Acinetobacter. So Acinetobacter is a genus of gram-negative bacteria belonging to the group Gamma Proteobacteria. Or so this particular Acinetobacter is a genus that is belongs to gram-negative bacteria, and it belongs to the group Gamma Proteobacteria. Acinetobacter species are non-motile and oxidase negative. Okay, so these are non-motile and oxidase negative and occur in pairs as observed under magnification. So when you observe under any magnifying devices like microscope, so you can see these bacteria are present in pairs. So young cultures show rod shape morphology. They are stick anaerobe that do not reduce nitrate. They are important soil and water organism and also found in many foods, especially refrigerated fresh product. Acinetobacter baumannii is a frequent cause of nosocomial pneumonia. So what is meant by nosocomial infection? So nosocomial infections, they are hospital-borne infection, and Acinetobacter baumannii is a frequent cause of nosocomial pneumonia, especially of late onset ventilator associated pneumonia. So it is serious pneumonia condition. It can cause various other infection including skin as well as wound infection and bacteremia and meningitis. So this is the first group of bacteria that is present in food, and this is Acinetobacter. And next it is Bacillus cereus. So Bacillus cereus is a thick, long, rod-shaped gram-positive, catalase-positive aerobic spore former, and the organism is important in foodborne illness. It is normally present in soil and is isolated from various foods also. So it is quite often cause of a diarrheal illness due to consumption of desserts, meat, dishes and dairy product, then rice and pasta, etc. So basically this Bacillus cereus is related to diarrheal infections after consumption of food like desserts, meat, as well as dishes, as well as some dairy products and some rice and pasta. Okay. So these foods are cooked and kept at room temperature as it is thermoduric. Okay. Some of the Bacillus cereus strains are cyclotropic as they grow at refrigeration temperature also. So Bacillus cereus is spread from soil and grass to cows, udders into raw milk. So basically this bacteria is present in raw milk, and if you process the milk improperly and you derive some product from that milk, obviously these bacteria will enter into that milk related product and it will cause harmful effects to the consumer who consumes that milk derived products. So it is also capable of establishing in cans. It is also capable of producing proteolytic and amylolytic enzymes, also phospholipase C, that is known as lecithinase. The production of these enzymes by these organisms can lead to spoilage of foods. So basically these microbes, these kind of microbes like Bacillus cereus, they produces proteolytic as well as amylolytic enzymes, and these enzymes when enter into your body after ingestion of food that is infected with Bacillus cereus causes severe illness. Okay. So diarrheal illness is caused by enterotoxin produced during the vegetative growth of Bacillus cereus in small intestine. So basically this bacteria Bacillus cereus, it induces diarrheal infection in a person after consumption of food that is contaminated with Bacillus cereus, and this diarrheal illness is due to enterotoxin that is secreted by vegetative strains of Bacillus cereus, and it is especially present in small intestine. So the bacterium has maximum growth and temperature around 48 to 50 degrees Celsius, and its pH range is 4.9 to 9.3. So like other spore mesophilic Bacillus species, spores of Bacillus cereus are also resistant to heat and survive after pasteurization temperature. So this is overall details regarding Bacillus cereus. So here you can show the picture that is showing Bacillus cereus. It is spore forming bacteria and it causes diarrheal infection in humans or the persons who consumes the food which is contaminated with Bacillus cereus, and the diarrheal infection are due to enterotoxin that is produced by this Bacillus cereus, and it has growth temperature of around 48 to 50 degrees Celsius, and pH range is about 4.9 to 9.3. Okay, so next is Bacillus subtilis. So Bacillus subtilis, also known as hay bacillus or grass bacillus. It is basically a gram positive and catalase negative positive bacterium commonly found in soil. It is a member of genus Bacillus. So Bacillus is genus also, and it is name of bacteria also. It is it is a genus also, and it is a type of shape also which is commonly found in bacteria. So Bacillus subtilis is a thin, short rod shaped and has ability to form tough protective endospore, allowing the organism to tolerate extreme environmental conditions. So basically this bacterium is present in soil and it forms endospore which is very tough and resist to harsh environmental conditions. So Bacillus subtilis produces proteolytic enzyme substilin, and the Bacillus subtilis spores can survive for extreme heat during cooking, and Bacillus subtilis is responsible for causing ropiness, a sticky, stringy consistency caused by bacterial production of long chain polysaccharide in spoiled bread dough. Okay, so a strains of Bacillus subtilis, formerly known as Bacillus nattois, is used in the commercial production of Japanese food natto as well as similar Korean food. Then it is used to produce amylase and also used to produce hyaluronic acid, which is useful in joint care sector in healthcare. So this is overall details regarding Bacillus, another bacterium that is present in milk. So basically this bacterium is present in soil and it produces spores that are resistant to harsh environmental conditions. So this bacterium causes ropiness in some foods. So this is how this causes foodborne illness. Okay. The next is Carnobacterium. So Carnobacterium is a genus of gram-positive bacteria within the family Leuconostosae, and Corynebacterium divergens and Corynebacterium bilateromachum are found in the wild and food products and can grow aerobically. So these species are not known to be pathogenic in humans, but it causes diseases in fish. So basically this bacteria is related to animals. It does not cause any harmful effects on humans. So the genus Carnobacterium contains 9 species, but only Carnobacterium divergens and Carnobacterium Baltaromaticum are frequently isolated from natural environments and foods. So they are tolerant to freezing as well as thawing and high pressure and able to grow at low temperature anaerobically. So they metabolize arginine and various carbohydrates including chitin, and this may improve their survival in environment. So Carnobacterium divergens and Carnobacterium Malta romaticum have been extensively studied a protective culture in order to inhibit growth of Listeria monocytogenes in fish and meat products. So several carnobacterial bacteriocins have been identified and described. So carnobacteria can spoil chilled foods also, but spoilage activity shows intraspecies and interspecies variation. So their production of tyramine in food is critical for susceptible individuals, but Cyanobacteria are not survive otherwise human pathogen. So these are overall details regarding Cyanobacterium. The next bacterium is Corynebacterium, which is commonly present in food. So Corynebacterium is a genus of gram-positive rod-shaped bacteria. So they are widely distributed in nature and are mostly innocuous. Some are useful in industrial settings such as Corynebacterium glutamicum. Other can cause human diseases, that is Corynebacterium diphtheria. So this is the main causative organism that causes foodborne illness, and it causes diphtheria. For example, in pathogens responsible for diphtheria, some species are known for their pathogenic effects in humans and other animals. Perhaps the most notable one is Corynebacterium diphtheria, which acquires the capacity to produce diphtheria toxin. So only after interacting with other bacteriophages. So diphtheria toxin is a single 60,000 molecular weight protein composed of two peptide chain, that is consists of two polypeptide fragment A as well as fragment B. They are together held by disulfide bond. So Corynebacterium diphtheria, it is mostly most causes diphtheria disease, and it is the main source of foodborne infection, which is commonly found. Then next is Clostridium perfringens. So Clostridium perfringens is a gram-positive encapsulated anaerobic non-motile bacterium. Encapsulated means it has capsule, and basically it is non-motile bacterium commonly found in meat and meat-related products. So it has ability to cause foodborne disease. So it is a toxin producing organism, produces Corynebacterium perfringens, enterotoxin and beta toxin that are active on human GI tract, that is gastrointestinal tract. So basically these bacteria are present in meat and meat products. So it multiplies very rapidly in food. Its doubling time is within 10 minutes. This bacteria get doubled if it is present in food and it get proper conditions. So spores are resistant to radiation, desiccation and heat, and thus survive in incompletely or inadequately cooked foods. However, tolerance is moderate to air. So vegetative cells of Clostridium perfringens are also somewhat heat tolerant as they have relatively high growth temperature. It obviously normally growth temperature for this bacterium is 43 degrees Celsius to 45 degrees Celsius, and can often grow at up to 50 degrees Celsius temperature. So they are not tolerant to refrigeration or freezing. So no growth occurs at 6 degrees Celsius. So Clostridium perfringens is present in soil and other natural environment. So this is the picture how Clostridium species look like. Then next, Clostridium botulinum. Again, it is important food pathogen. So Clostridium botulinum produces most potent toxin known as botulinum toxin. So it is a gram-positive aerobic rod-shaped bacterium. It is oval. It produces oval endospores that are produced during stationary phase. In cultures, there are 7 types of Clostridium botulinum, that is from group A to G, based on serological specificity of neurotoxin they produce. So these are 7 groups that are categorized based on serological properties, and these serological properties, they are mostly based on neurotoxin they produce. So botulinum is a rare but a very serious disease. The ingestion of neurotoxin produced by the organism in foods can lead to death. So however, the toxin, a protein, is easily inactivated by heat, and the organism can grow at temperature ranging from 10 degree to 48 degrees Celsius with an optimal growth temperature at 37 degrees Celsius, and spores are highly heat resistant. The outgrowth of spores is inhabited below at pH 4.6 and salt concentration of high salt concentration. Okay. So basically this Clostridium botulinum, it produces harmful toxin, and it is neurotoxin. This toxin affects on your nervous system, and it is a very severe disease, foodborne disease, and it causes death. Okay. So botulinum spores are probably the most radiation resistant spores of public health concern. So contamination of food is through soil and sediments where they are commonly present. So this bacterium mostly present in soil and sediments, and the contamination occurs through these modes. So the organism grow under obligate anaerobic conditions and produces toxins under processed or improperly canned low acidic foods at ambient temperature. So this is overall details regarding Clostridium botulinum. The next bacteria is Campylobacter. So Campylobacter are gram-negative non-spore forming rods. Campylobacter jejuni is an important foodborne pathogen. So it is one of many species within genus Campylobacter. So Campylobacter species, that is Campylobacter jejuni, Campylobacter coli, cause diarrhea in humans, and the organism is sensitive. It destroys after pasteurization of after at pasteurization temperature. It is also sensitive to freezing. So these organisms belong to family Campylobacteriaceae, and the organism they are basically curved or S-shaped or spiral rods, and may form spherical as well as coccoid forms in old culture and culture exposed to air for prolonged periods. So most of the species are microaerophilic. It is oxidase and catalase positive, and do not grow in presence of 3.5% salt or at below 25 degrees Celsius or below. So the incidences reported gastroenteritis by an organism at high in case of Salmonella. So basically this Campylobacter jejuni is important foodborne pathogen, and it leads to foodborne diseases like diarrhea and gastroenteritis. Okay. So this is a picture. It is basically a S shape or curve shape bacteria. Okay, and this is picture that shows how this bacteria look like. The next bacteria is Erwinia. So Erwinia is a genus of the family Enterobacteriaceae bacteria, containing mostly plant pathogenic species. So this group of bacteria, they are mostly plant pathogens. So the organism was named after first phytobacteriologist, that is Erwin Smith, and it is gram-negative bacterium related to E. coli, Shigella, Salmonella, and Yersinia. It is basically a member of Enterobacteriaceae family. So it is primarily a rod-shaped bacterium. A well-known member of this genus is species Erwinia amalowhora, which causes fire blight on people and pear and other rosaceous crops. So Erwinia coroteva, also known as Pacobacterium corotivecum, is another species. It causes diseases in many plants, and this species produces pectolytic enzymes that hydrolyze pectin between individual plant cells, and decay is caused by Erwinia caratova, is often referred as bacterial soft rot (BSR), and most plants or plant parts it can resist invasion by bacteria unless some type of wound is present. So high humidity and temperatures around 30 degrees Celsius are favorable conditions for development of this bacterium and development of decay. Then next bacterium is Enterococcus, that is Enterococcus faecicum and Enterococcus faecalis. So Enterococcus is a genus of lactic acid bacteria. So Enterococci are gram-positive cocci that are often found in pairs, that is diplococci or short chain, and are difficult to distinguish from streptococci on physical characters mentioned above. So the two species are commensal organism in intestine of humans. So lactic acid bacteria, they are mostly the part of our normal flora of our body, and lactic acid bacteria are considered to be the good, but there are some species of lactic acid bacteria, especially enterococcus, so they are pathogenic. So enterococci are facultative anaerobic organisms. They are mostly non-spore forming bacteria, and they grow optimally at 35 degrees Celsius. However, they tolerate a wide range of environmental conditions, just like growth temperature that is up to 10 to 45 degrees Celsius, and pH range for these bacteria is almost 4.5 to 10.5, and they can even tolerate high salt concentration up to 0.6-0.5%, and can survive in heating at 60 degrees Celsius for 30 minutes. So these are catalase negative, oxidase negative bacteria of genus Enterococcus. So these are ubiquitous organisms that are found occur in large number on vegetables, then plant material and food, especially those of animal origin such as meat and dairy products. So Enterococci also constitute large preparations of autochthonous bacteria associated with mammalian gastrointestinal tract. So the resistance of enterococci to pasteurization temperatures and their adaptability to different substrate and growth conditions in food products manufactured from raw material and in heat treated food product is one of the great significance. So enterococci may constitute an important part of microflora of fermented food as well as meat and cheese. Okay, so this is overall details of enterococcus. Then next is Escherichia coli. So E. coli strain are associated with foodborne gastroenteritis. Okay, so E. coli is again one of the important pathogen that is present in food, and it causes disease that is known as gastroenteritis. So it is a gram-negative asparaginous rod that ferment lactose and produces dark colonies with metallic sheen on endoagar. The organism grows well on a large number of media and in many foods, and they grow over a wide range of temperature, that is 4 degree to 46 degrees Celsius, and pH range of 4.4 to 9.0. So however, they grow very slowly in foods which is stored at refrigerating temperature, that are 5 degrees Celsius, and they belong to family Enterobacteriaceae. The organism is also an indicator of fecal pollution. These bacteria of Enterobacteriaceae family, and these are mostly popularly known as coliform bacteria because they cause fecal infection. Okay, so the organism also capable of producing acid and gas and of flavors in foods. So E. coli strains are involved in foodborne illness can be placed into 5 groups, that are enteropathogenic, then enterotoxigenic, then enteroinvasive, enterohemorrhagic, and facultatively enteropathogenic. So all these are 5 groups of Enterobacteriaceae family, and all these are they causes foodborne infection, and these are important food pathogens. So the organisms also grows in presence of bile salt. So the primary inhabit of E. coli is intestinal tract of most warm-blooded animals. So basically this E. coli bacteria is present in intestinal tract of mostly warm-blooded animal, and there are different strains of E. coli bacteria. Okay, so the strains are some strains are usually tolerant to acidic environment. So this is a picture showing E. coli cells. Then next is lactobacillus. So this lactobacillus is a genus of lactic acid bacteria that were formed, including genus Streptococcus, and they are known as homofermenters, meaning they have produced a single product of glucose fermentation. So they are gram negative, they are gram positive, catalase negative, non-motile occurs that are found singly in pairs or in chains. Some of the strains of lactococci are known to be grow at or below 7 degrees Celsius. So lactobacillus is commonly found in milk, okay, and they are mostly involved in lactic acid fermentation processes that are carried out in milk as well as milk related other products. So lactococci are intimately associated with dairy product. So these organisms are commonly used in dairy industry in manufacture of fermented dairy products like cheese. So they can be used in single strain starter culture or in a mixed strain starter with other lactic acid bacteria such as lactobacillus and streptococcus species. So their main purpose in dairy production is rapid acidification of milk. So mostly these bacteria are used in milk fermentation and fermentation of other milk products. So this causes a drop in pH of fermented food, which prevent the growth of spoilage and pathogenic bacteria. So these bacteria also play a role in flavor and final product. So dairy lactococci have also been exploited for several industrial fermentations and several other biotic industries. So they are easily grown at industrial scale up on cheap whey based media. So these strains of lactobacilla they are mostly used in industry, various industries just like food industry as well as biotic industry, because they are easily grown on different kinds of media, especially cheap media. So lactococcus lactis subspecies, lactis includes species formerly designated as streptococcus lactis and lactis. So lactis subspecies cremoris is distinguished from lactobacillus lactis subspecies. So lactis by inability, that is it grows at 40 degrees Celsius, grows at 4% NaCl concentration, then it hydrolyzes arginine and ferment ribose. So these are biochemical details of identifying different lactobacillus or lactic acid bacteria species among themselves. Then next is lactobacillus. It includes lactobacillus vulgaris, then lactobacillus helveticus, then lactobacillus plantarum. Okay, so these organisms belong to important genus and rods, usually long and slender, and in some of the species they form chain, and they are aerotorrent microaerophilic, but some ferment sugars chiefly to lactic acid if they are homofermentative. The heterofermentative species besides lactic acid all produces small amount of acid, then carbon dioxide and trace amount of volatile compounds such as aldehydes and alcohols. So the homofermentative species of lactobacillus includes Lactobacillus bulgaricus, Lactobacillus cz, then Lactobacillus helveticus, then Lactobacillus lactis, and Lactobacillus acidophilus, and they grow optimally at 37 degrees Celsius. So these are pretty much important bacteria as far as fermentation, whether it is homofermentation or heterofermentation are concerned. So lactobacilli are of considerable importance in food as they ferment sugar to lactic acid and other desirable flavoring compounds, and are thus used in production of fermented dairy products as well as meat products. However, they are also implicated in spoilage of wine, in beer. So these are both harmful as well as beneficial. They shows both harmful as well as beneficial activities as far as this food industry is concerned. In case of food, they are beneficial, but in case of beverages like wine and beer, they causing souring of wine and beer by producing lactic acid. So these organisms normally occur in plant surface silage, then they occur in manure and dairy products. So they are quite fastidious in their nutritional requirement, as they are unable to synthesize certain vitamins they require, and therefore media need to be supplemented with these vitamins for their growth. So these are growth conditions that are important for lactobacillus or lactic acid bacteria. Some of the strains of some of the strains are psychotropic in nature and are thus involved in spoilage of refrigerated foods and meat. On the other hand, thermodynamic properties, that is resistance to pasteurization temperature of some of the thermophilic strains of lactobacilli, are quite useful in manufacture of certain varieties of cheese, example Swiss cheese. And some of the strains of lactobacilli also show probiotic attributes and are finding application in functional probiotic foods as well as pharmaceutical preparation. So probiotics is the area. So these are good bacteria, and mostly probiotic bacteria includes some of the species of lactobacillus or lactic acid bacteria. Then next bacteria is leuconostoc. So leuconostoc is especially present in it is used in fermentations of various products. So leuconostoc is a genus of gram-positive bacteria. It is placed within family Leuconostaceae, and they are generally ovoid cocci, often forming chains. And leuconostoc species are intrinsically resistant to vancomycin and are catalase negative, which distinguishes them from staphylococci. So all species within this genus are heterofermentative and are able to produce dextran and sucrose. So they are generally slime forming, blamed for causing sting when creating a soundruff starter, and some species are also capable of causing a human infection. So leuconostoc species along with other lactic acid bacteria such as Pediococcus and Lactobacillus species is responsible for fermentation of cabbage to sauerkraut, and in this process the sugars in fresh cabbage are transformed to lactic acid, which gives a sour flavor and good keeping qualities. Then next bacteria is Listeria monocytogenes. So Listeria monocytogenes is in food has attracted worldwide attention due to a serious illness it causes in human beings. So Listeria are gram-positive, non-spore forming, non-acid food fast food rods. Okay. So the organisms is catalase positive, and products it produces lactic acid from glucose and other fermentable sugars. The organism grows well in brain heart infusion, trypticase soya and trypticase broth. However, the medium should be fortified with vitamin B and some amino acids. It is a mesophilic organism with optimal growth temperature at 37 degrees Celsius, but it can grow at refrigerated temperature also. The strains grow over temperature range from 1 degree Celsius to 45 degrees Celsius and pH range of 4.1 to 9.6. So again, Listeria monocytogen, it is important foodborne pathogen, and it causes severe health effects after getting to your body after consumption of food that is contaminated with Listeria monocytogen. So Listeria monocytogenes is widely distributed in nature and can be isolated from decaying vegetation as well as soil and animal fecus. Then you can isolate the same from sewage, silage as well as water. So the organism has been found in raw milk. It is pork raw. You can found this bacterium in raw milk as well as pork or raw poultry and ground beef as well as vegetables. The high temperature short time treatment of pasteurization is good enough to destroy this organism in milk. So the most significant viral factor associated with this bacteria is Listeria-lysine O. The virulence strain produces beta hemolysis on blood sugar and acid from rhamnose. So Listeria monocytogenes grow well in moderate salt condition or moderate salt concentration, and Listeria monocytogen in unique among the foodborne pathogen, while other pathogens excrete toxin or multiply in the foodborne stream. Listeria monocytogenes enters into the host cell and grows inside the cell. So in humans, it crosses the intestinal barrier after entering by oral route, and it causes severe infection to humans. Ready to eat foods that are preserved by refrigeration pose a special challenge with regard to Listeria monocytogen infection. Okay, so these are overall details regarding important foodborne pathogen, that is Listeria monocytogens. Then another So this is sufficient for this lecture. There are a lot of bacteria and organisms we have to study. So remaining microorganisms and bacteria will study in next lecture. So far, thank you.